Okay, the first step in electrophoresis is to prepare a gel. So when we run our gel electrophoresis, we're going to run our gels in what's called TBE buffer. Okay, so what I do is I order a 10x TBE buffer from a molecular biology company. I basically then take that 10x TBE buffer and dilute it 1 to 10. So what I do is I take one of these containers, I add 450 mLs of deionized water, and then I add 50 mLs of my 10x TBE buffer. So here's my buffer solution all ready to go. When I make up my gel, then what I want to do is I want to make up my gel in TBE buffer. So I'm going to make up 100 milliliters of my agarose gel and TBE buffer. So this is agarose. Agarose is basically a fine powder. That powder then is like gelatin. Okay, the key thing, you can actually run electrophoresis in gelatin. And there are a lot of home lab curricula where if we did want to do this at home, you could set up an electrophoresis unit by taking batteries, taking leads, making up agarose, and then using a touch of salt in salt water, and then run your own electrophoresis. The hard part, of course, is coming up with the wells, which you'll see in a second. So what I'm going to start with then is I'm going to add, if I can get this open, I'll add 100 mLs of our TBE buffer. So now we have 100 mLs of our TBE buffer. And this container, give or take a few, doesn't matter too much. I'm then going to make a 0.8% solution of agarose. So 0.8% means I'm going to take 0.8 grams of agarose, place it in this container. So I'll put on my weigh paper, tare, take my agarose, and we're looking for 0.8 grams of agarose. And when you run electrophoresis, we can go anywhere from 0.8 to 1%, even 1.2%, still get good results. So I'm not going to worry about being exactly right on. So there's 0.82 grams of agarose. You can see agarose is a nice fine little powder right there. So I'll take that powder, add it to my solution, spin it around a little bit. Okay, I'm then going to add an aluminum foil cap. Well, actually, I won't add aluminum foil cap because I'm putting this in the microwave. I will then basically put this in the microwave for a few minutes to melt the agarose so that I have a fully melted solution before I pour it into the molding tray. Okay, so here is my agarose and TBE buffer. Had a little bit boil out because it was hot here, so we're a little slightly less than 100. So I stir it around because agarose will go down to the bottom. Over here I have my molding tray. So here's my electrophoresis tray right here. I'll put that in and then tighten these up so that it's all ready to go. Okay, so these then tighten up both ends. My tray is open on this end and this end, so I have to make sure it's tight there. This is what's called a comb, so this is going to make little wells in my mold. I put my comb right there. Then I take my agarose and I pour it so that it's just a little ways up the bottom of the comb so that when it solidifies it makes those wells. Now we leave it to solidify for about 20 minutes. 20 minutes have passed and you can see how the agarose has solidified. So in here it's nice and solid. You kind of see how that's like gelatin in there. Okay, the same thing has happened inside my tray. So what I'm going to do now is I'm going to gently take this comb out. So I've got to gently pry it off of those wells. And then you can see the gel here. Here's our gel. Okay, here are all of our wells. And we're then going to use the gel in gel electrophoresis. All right, the next step then is electrophoresis. So we have the gel that we just made right here. We have where our combs were, and we have the agarose gel there. So I'm going to take the lid off of this. This is our electrophoresis unit. We're going to put the gel into the electrophoresis unit. Okay, and so in the gel, since DNA is negatively charged, it's going to go to the positive electrodes. So what we want to do is run to red. So if these are our wells, we want our wells on the side where the black electrodes are, and we want to run to red. So I'm going to put the electrophoresis tray inside the unit with the wells on the opposite side of the red. Then we're going to take the TBE buffer, and we will add the TBE buffer to the tray, carefully filling it up right up to the top edge. We'll add this to the tray, filling it right up to the very top edge on this side as well. Then I'm going to go barely over the top. Okay, so it goes barely over it. All right, so now we're set there, and we're ready to move on to the next step. So the next step is to basically add our samples to the electrophoresis gel. So here's a sample. It's in a microcentrifuge tube. That microcentrifuge tube then is labeled. This one is sample A. This one's going to go in the leftmost well, and going right, each well number 2 will be sample B, 3 sample C, 4 sample D, next one sample E, then Sophie, Donna, Bill, Harry, Sam, Frank, and finally in the last well will be the ladder. Okay, so the next step is to fill the wells. So I have a pipetter. This pipetter is set to 10 microliters. I'm going to place 10 microliters of each sample into the wells. So the first thing I'm doing is getting a pipette tip. I'm then going to get the sample. This is sample A. I shall fill the sample into my pipette tip. And then I have to just make sure that I get this down nice and even right in the well. So I'll have to steady my hand in the well. Then I go down all the way and pull that out. Okay, so that one's filled. I'm going to switch pipette tips, grab the next sample, and I'm going to fill this sample. You'll be able to see this one a little bit better because this one is red. Then I'm going to steady my hand, go through, and fill that well. So you can see those wells are filled there. Okay, might as well do a third one while we're at it, just so that you can see this one more time. Change pipette tips. So the third well over is going to be so I'm going to continue filling then all wells as we said before. Alright, now all our wells are filled. Now it's time to just put on the lid, hook up our electrodes, and then set it up to the power supply. So put the lid on, hook it down, and our power supply is right here in the back. And I'm going to turn it on and start the electrical current. Alright, now the power supply is on. I changed the angle a little bit, and so we can start seeing how the dyes are starting to move up that electrophoresis gel. So they're moving then to the positive electrode, and they're negatively charged and moving toward that positive electrode. And you can see them moving quite a bit there. Now dyes move a lot faster than DNA, so if this was DNA, this would take about an hour. Whereas these dies, then I'll be able to move them up to about the right position in about 15 to 20 minutes. And if this was DNA, you wouldn't really be able to see anything because the DNA is colorless unless we use a fluorescent dye so that we're able to visualize it. In this case, the dyes all have, are all capable of being visualized as they move. So you can kind of see that movement of these dyes, which makes it a little bit more fun to run. Okay, that's looking pretty good now, and so we'll go ahead and turn this off. And then take the lid off so we can see that in there. Okay. So you can see that nicely in there. Alright, and then we can kind of talk about all of this stuff. Okay, so well 1 over there is sample A, sample B, sample C, sample D, sample E. Then it goes, after that, then Sophie, Donna, Bill, Harry, Sam, and then Frank. I don't have a ladder in here. And so if there is a ladder, then the ladder has a blue one being the closest. There's a green that none of these have, being between the blue and the purple. So it goes blue, green, purple, red, yellow. So you can see 5 different ones there. There is no green. Then each person has up to 2, and I'll give you a hint, every single person here has 2. And so you can just start, right, this one's a red, yellow, and you can kind of move down and figure out who is what with this. It looks pretty clear.